Welcome back to the Basic Bioinformatics Web Workshop. In this lecture, we will focus on the fundamentals of sequence alignment. We will start by discussing general properties, then we will try to understand the calculations and evaluation of alignments using an example. Finally, we will look at the differences and possible applications of different algorithms. Before we begin this lecture, let us remember what we should have learned from the previous one. To perform a sequence alignment, a minimum of 2 sequences is required. Amino acid sequences can be retrieved from UniProt, while nucleic acid sequences are recommended to be found at NCBI. However, both types of sequences can also be found in other databases, like EMBL for example. When I was first introduced to the topic of sequence alignments and sequence analysis in general, I had a lot of questions going through my head regarding those topics, and I thought maybe it would be a great idea to introduce you to the topic by targeting those same questions that I asked myself previously, since you might be on the same level as I was back then. So the primary question you are probably asking yourself at the beginning of this lecture is, why is sequence analysis necessary? Or what are these alignments anyway? And how do they relate to sequence analysis? In short, the purpose and goal of sequence analysis and alignments are to identify similarities and differences between DNA or amino acid sequences in diverse organisms. Sequence alignments are the most common task in sequence analysis. The ultimate purpose of sequence alignments is to measure sequence similarity in between a pair or even sets of sequences. By aligning sequences, it is possible to identify regions of similarity indicating potential functional importance. For sequence analysis, aligning sequences serves various purposes. First being the predictions for functions of genes or secondary structures that can be made through aligning your sequence of interest against a reference sequence or a database. In fact, alignment algorithms are commonly utilized in most databases as search algorithms for the purpose of query searches. Gene finding can also be done by aligning a reference sequence against a genome, so you can determine whether and where the potential gene that you're searching is present in your genome. Alignment strategies are used in sequence assemblies to arrange DNA fragments, as it is often required after sequencing. However, alignments serve an important purpose in identifying divergence and similarity between 2 or more sequences. This can give us an indication on their evolutionary relationship. Now that we understand the capabilities of sequence alignments, it may still be difficult to visualize what an alignment would look like. Well, let's take a look. So, as shown in the table, alignments can be constructed in various types. On the left side, there is a sequence alignment of 3 partial DNA sequences over a common length of 23 base pairs. As long as there are more than 2 sequences involved, we are talking about a multiple sequence alignment. Conversely, on the left side, we observe a pairwise amino acid alignment. In pairwise alignments, only 2 sequences are aligned. Beside the number of involved sequences and their biochemical properties, there is another difference that can be observed. The protein alignment results in a 3-digit gap after the 4th letter. This is what we refer to as a gapped alignment. So we have an gapped alignment and an ungapped alignment. An observant student may notice that the protein alignment has another gap. However, the gap between U and U is due to the fact that the letter U is not used in the one-letter coding of the amino acids. Don't worry, this fact has been ignored in the following examples, as it is not relevant for the explanations of the principles. What really is important, however, is that in addition to the three more obvious differences already described, there are other factors that ultimately lead to distinct alignments. First being that alignments can be viewed over their entire length or in regions of high similarity. The choice of alignments for determining the optimal alignment is critical and can lead to a wide range of different results. Another aspect included in the calculation, which plays a significant role in the final result when considered individually, is the individual evaluation of matches, mismatches, and gaps. Before we go into more depth on the individual aspects, we should first clarify how we recognize a meaningful alignment. The fundamental question when conducting an alignment and visualizing the results might be whether the perceived similarities among the sequences happen due to chance or hold a fundamental biological significance. While observing similarities among sequences, one may assume a corresponding degree of homology. However, in reality, this is not always the case. The terms homology and similarity are frequently used interchangeably. However, each bears a unique significance. Similarity is a descriptive term indicating some percentile degree of match within the compared sequences. Homology, in contrast, has clear evolutionary and biological implications. Within the context of molecular biology, it is specifically referred to as similarity in sequences or structure due to descent from a common ancestor. Thus, over time, the accumulation of mutations is the cause of the divergence between the compared sequences, as well as in comparison to their last common ancestor. In conclusion, the determination of homology between sequences is a binary outcome that can be answered with yes or no. Similarity, however, can be defined quantitatively. For instance, one could observe that both sequences share an 80% similarity. However, it would be incorrect to claim that homology of the sequences is 80%, even though high similarity is a good evidence for a common recent ancestor and hence homology. If two sequences are homologous and a similarity between them is only reflected in certain segments, then these segments are referred to as conserved regions. When assessing alignments, it is crucial to provide an objective representation of the observed outcomes and their precise labeling, particularly in cases where correlations in sequence or structure are not obvious due to convergent or divergent evolution. If you are now asking yourself, what was convergent and divergent evolution again? Divergent evolution is a classic model where over time, due to mutation and selection, the different descendants become more and more different from the common ancestor. Convergent evolution is the model of the independent evolution of similar features in species due to external pressures. This does not only apply for the evolution of similar features in higher organisms. These mechanisms has also been demonstrated in protein structures that share common 3D folds, but nowhere near common ancestors. Convergent evolution does not usually produce highly similar sequences of any great length. However, the same may be true for some homologs. In this case, an alignment can only lead to a hypothesis regarding homology, but itself does not imply an evolutionary order of events without any additional information. Now that we have clarified the terms of homology and similarity, I would like to return to our previous slide once again. The table represents 3 out of the 6 attributes mentioned, including protein and DNA alignments, pairwise and multiple sequence alignments, and gapped or ungapped alignments. These are the more obvious properties that can be determined right away. However, it is more challenging to distinguish among the other 3 attributes because they also affect each other. For instance, it's difficult to identify the algorithm used in a particular alignment through pure visualization. On the other hand, differences between alignments of the same sequences are recognizable when using different algorithms. Things get a little bit more complicated here, as you can see. So we need to ask ourselves what the overall goal might be when aligning A and B, for instance, in a pairwise alignment. Simply, we want to compare 2 sequences against each other and find the best possible alignment between them, also known as the optimal alignment. Even though the answer might sound simple and the example might seem easy, there's much more to it than that. If we enhance our example from 15 prefixes to 50 or even 500, finding the optimal alignment between both A and B would be much more difficult. This is where the optimal alignment algorithm comes into play. To determine a mathematical accurate algorithm, it might be helpful to visualize our problem at first. This can be done by something called dot plot matrix. All prefixes of both sequences are displayed on the axis of the matrix. By comparing each prefix of both sequences directly, all positions with a match are marked with a dot. Such matrices are one of the simplest ways to compare sequence similarity graphically. However, this method can suffer from background noise, as you can see in A. To distinguish a meaningful dot plot pattern arising from the background noise is usually applied by the filter. The most common filtering technique uses filtering windows of fixed length, requiring a comparison to meet a minimum identity score summed over that window before being considered. So only diagonals of a certain length will survive the filter, as shown in B. The displayed example shows the same sequence aligned against itself. After we have visualized our problem, we need 2 more things to solve it. In order to achieve an optimal alignment, we first need an algorithm to produce the results, as well as an evaluation to score the results. If we look back at our dot plot matrix, we see either a dot or an empty space. A dot indicates a match, and a space indicates either a mismatch between the 2 prefixes or the presence of a gap on either axis. So our evaluation could be the following. For each match, we score a +1. For each mismatch or gap, we score a -1. This is basically what is described in a formula. Sigma has a value of -1 if the prefix A in the first sequence is matched with a gap or vice versa for sequence B. Sigma also has a negative value if the logic between A and B is not identical. In case of a similarity between A and B, sigma would have a positive value here as +1. When we are researching algorithms for sequence alignments, then there are 4 names that will come up immediately. That's being Needleman and Wunsch, as well as Smith and Waterman. As for our first example, I will introduce the Needleman-Wunsch algorithm. Do not worry, we will come back to the other algorithm at a later point. The Needleman-Wunsch algorithm calculates the maximum score for all possible residue pairs between sequences S and T over the total length of both. This score is represented in the formula as Mij. The elements I and j stand for variables for the single residue within the sequence S and T, respectively as Si and Tj. For those of you who are confused, S and T are just individual terms for the example sequences, which were A and B in the previous example. So S2 is the second residue from the sequence S and corresponds to an H in the example. The resulting matrix of the two sequences is similar to the dot plot matrix, except that the spaces and the dots are filled with the individual scores for each position. The matrix has a size of n times m, which is defined by the length of both sequences. However, the first or zeroth position, m00, always starts with a gap and the value 0. Accordingly, the first row and column of the matrix results in negative scores only, since S and T are evaluated against the gaps, starting from the 0th position. This evaluation of the matrix boundary is called initialization. The algorithm then recursively calculates the individual values of the position based on the previous position. As a result, all positions in the alignment influence each other. To understand this better, I will now show you how the scores are calculated using the first four residues. The maximum value for position M11 can result from three possible origins: a vertical move resulting from the top value, a horizontal move resulting from the left value, or a diagonal move resulting from the origin. A horizontal move, therefore, means a gap in sequence S. A vertical move results in a gap in sequence T. A diagonal move corresponds to a match or a mismatch. This relation is generally valid. Specifically, this means the following for our position. The vertical move achieves a negative value of -1 due to the gap as previously defined, which adds up to the score of the original position, resulting in the score of -2. This makes sense because two consecutive gaps must achieve such a value if we apply our previously defined logic that a gap achieves a value of -1. Accordingly, the horizontal movement must achieve the same result. Starting from the origin, a score of 0, S1 is matched against T1 in a diagonal movement. As both positions correspond to a T, there is a match which adds the value +1 to the previous score of 0, which ultimately results in the score of +1. That is the maximum score for the position M11 compared to the other two possibilities. Using this method, we can now calculate the scores for all surrounding positions. In order to calculate the maximum score for M22, we must first calculate the scores M12 and M21. M21 is the position to the right of the previously calculated value of M11. A vertical movement with a further gap would result in the value of -3. As a diagonal movement corresponds to a mismatch of H and T, the value of the cell would be -2. The horizontal movement results in a gap of the sequence T resulting from M11. Since this position is reduced by -1, the maximum score for the position M21 is 0. The same result applies to the position M12 below M11, except that a horizontal movement is necessary due to the different starting point. The horizontal, vertical, and diagonal neighboring position to M22 is now scored, so a calculation of M22 can be conducted. A gap caused by a horizontal or vertical movement would result in a score of -1. Since the second position of both sequences corresponds to an H, the value +1 is added to the existing score of M11, resulting in a maximum score of 2 for the position M22 due to a diagonal movement. If this procedure is applied to every position within the matrix, the result would be the follows. Now that we have completed the initialization and the scoring of the matrix, there's still one thing missing. We still do not have a finished alignment. So far, we have only a bunch of numbers in a grid. The third step, which translates the numbers into an alignment, represented by letters, is called the traceback. The last position at the bottom right of the matrix represents the optimal alignment score. Starting from this, we move backwards in our alignment to the origin M00. As already mentioned, each diagonal movement on this path corresponds to a match or mismatch between the positions of the two sequences. A vertical step corresponds to a gap in the upper sequence. A horizontal step stands for a gap in the left sequence next to the matrix. If we apply this method to our alignment, we notice that starting from the maximum alignment score at the bottom, the path runs exactly diagonal. However, after position M58, the situation is not longer as clear as before, as the middle section represents several possible paths. This means that more than one optimal alignment is possible under the given circumstances. The remaining path from M22, which we discussed in detail earlier, is again diagonal. So accordingly, the following 4 alignments with the max score of 9 are the results of our efforts to align these sequences. Now that we have shown you how to perform an alignment using the Needleman-Wunsch algorithm, we need to answer another question. Earlier I talked about the optimal alignment. So why is there more than one optimal alignment? Is there even a single best alignment? The answer is yes and no. I mentioned earlier that the alignment is computed by an algorithm and an evaluation of the results. The Needleman-Wunsch algorithm used in our example was not the deciding factor. It was actually our trivial evaluation. The choice was made for its pedagogical value, not for its scientific correctness. This may be the reason why we have 4 different alignments with the same optimal score. To answer our question, because of the evaluation parameters we chose, it was not possible to compute a single optimal alignment using the 2 example sequences. For this reason, researchers use more complex evaluation parameters. The most common tool for this purpose is the use of a substitution matrix. This tool assigns individual scores for each match or mismatch. There are a wide variety of matrices to choose from, and many modern algorithms and software solutions already include them. Obviously, there are different matrices for amino acids and nucleic acid sequences. However, differences between matrices for protein sequences can be based on theoretical considerations, such as the number of mutations needed to convert an amino acid or the physiochemical properties of the compared residues. Not all amino acid pairings are evaluated equally. The evaluation takes into account both the statistical frequency of the pairing and, in case of a mismatch, the probability of the mutation. The exchange of amino acids with similar character, for example isoleucine and leucine, occurs more frequently in nature than that of amino acids of different character, like isoleucine and aspartic acid. There's no single best universal matrix, and the selection of these parameters can vary depending on the circumstances and goals of the research. For example, we would not want to use a matrix that heavily penalizes mutations for an alignment that aims to show consensus areas in more distant relationships, or vice versa. There are two main groups of protein substitution matrices, the PAM and Blosum matrices. The Blosum matrix, derived from clustered experimental data extracted from a collection of multiple sequence alignments. Blosum is always represented by a number, for example Blosum 62. This number is associated with the matrix values. Higher matrix values correspond to a more conservative evaluation, so that Blosum 90 would be the preferred fit for a study of closely related sequences, whereas Blosum 45 would be preferred for distantly related sequences. Blosum 62 is more or less the medium fit, and it's therefore often the default substitution in modern alignment tools. The PAM matrices are based on observed amino acid changes in closely related sequences, assuming a constant mutation rate. In terms of matrix values, it is the opposite of Blosum, with PAM1 representing the most conservative assessment, down to PAM 250, which is more divergent. In conclusion, both Blosum and PAM differ in their derivation methods, scoring interpretations, and applications. Blosum matrices are more commonly used in local alignment algorithms, while PAM matrices are applied to global sequence alignments based on the constant mutation rate assumption. Another important parameter for evaluating the alignment is actually the part that does not even align. I'm talking about the gaps. Gaps usually occur for two reasons. If you're looking at a pairwise alignment of two homologous sequences, either one of them has had a mutation that shows a deletion or an insertion of a residue at the given position. Since it's not always clear or important whether it's an insertion in sequence A or deletion in sequence B, these regions are commonly referred to as indels. In our previous example, we used a gap penalty of -1 for each indel. This linear approach to a gap penalty is common. However, it can be problematic if it's not properly balanced. If the penalty is too low, more gaps will occur, which could lead to a meaningless alignment that does not show a realistic relationship even though its score is maximized. The problem is that studies of the functional structure of altered sequences have shown that indels often occur over multiple residue long segments. If the gap penalty is too high, the alignment would again not show a realistic relationship between the sequences. In addition to the linear approach, dynamic models are also used to fit the observed evolutionary events. These models penalize the first indel more heavily than subsequent indels. For instance, the gap opening penalty would be -5, while each following gap extension penalty would be -1. Just as with the selection of the appropriate substitution matrix, the selection of the gap penalty has a significant impact on the resulting alignment and should be considered on a case-by-case basis. It is important to keep in mind that alignments and their evaluation are merely models and as such are not perfect representations of real relationships. Nevertheless, they are a useful tool that aid in our understanding and examination of these relationships. With that being said, I would like to finish the aspect of evaluating alignments and once more return to one of our previous slides. When we were discussing how to score an alignment, I proposed the idea that we need two things. First being the evaluation and second being the algorithm. Now that we have talked in depth about the evaluation, let us now focus on the algorithm. In addition to the Needleman-Wunsch algorithm that we used in our example, it is important to be familiar with the Smith-Waterman algorithm. The Smith-Waterman algorithm is a modification to the Needleman-Wunsch algorithm, and it is used for local alignments. It compares sequences of all possible lengths and selects the most conserved segment based on the maximum score. As a result, the output is not an end-to-end alignment, but rather the most conserved region among all compared sequences. The prefix and suffix regions of the sequences that do not share a high similarity are excluded from the alignment. Local sequence alignment is useful when searching for a domain or a motive within larger sequences. The algorithm follows the same steps as a Needleman-Wunsch, but with two main differences. First being the calculation of the max scores. Like in our previous example, we have selected a scoring function that assigns negative values to mismatches and gaps and positive values to matches, where the scoring function is additive. The same recursive relation as before is used, but a fourth option is introduced. Whenever the cell would normally take on a negative value, it is instead assigned a value of 0. This ensures that the score cannot become negative, making matching segments stand out from the surrounding. The second difference concerns the traceback. It starts with the highest value of the matrix, and the path is followed, similar to Needleman and Wunsch. However, unlike Needleman and Wunsch, the maximum score is not always located in the bottom right corner of the matrix, and the path ends as soon as a score of 0 is reached. As a result, this procedure produces shorter, more conserved segments compared to longer global alignments. If this might have sounded a little bit complicated, I will just show you a quick example. Both sequences A and B were aligned using the same scoring for both algorithms. The results differ in length and score as expected. The TAAT motive was aligned by the Smith-Waterman algorithm, while the Needleman-Wunsch algorithm aligned the sequences over their full length. I hope the visualization helped you to understand the concept. Okay, now that we have talked a lot about differences, I would like to highlight one thing both algorithms have in common. They both produce an optimal alignment. If you might remember our first lecture of the series, in case you have watched it, we were talking about databases and web tools. Now I would like you to imagine the following problem. Our database contains more than 100 million individual sequences, and each ranging from a few residues to several millions. So how are we going to compute an optimal alignment using the methods we introduced earlier? The answer is simple. It is not possible. It is a computational problem that we cannot overcome. This leads us to a follow-up question. How are we even able to search against a database with our query sequence to find any certain degree of match or even matches? In the 90s, someone called Stephen Altschul and his colleagues found a solution to this problem, which is now known as BLAST. The BLAST algorithm is also a local alignment algorithm, like the Smith-Waterman algorithm, but there are some important differences between them. Both the Needleman-Wunsch and Smith-Waterman algorithms solve problems by breaking them down into smaller, overlapping subproblems, solving each subproblem only once, and storing the solution to avoid redundant computation. This method is commonly referred to as dynamic programming. BLAST is not based on dynamic programming. It uses a heuristic approach. While dynamic programming guarantees an optimal solution, it can be computationally expensive, especially in larger datasets. Heuristic approaches, such as those used in BLAST, aim to provide faster solutions. Simply said, accuracy or optimality is sacrificed for efficiency. To compute an alignment with BLAST, the algorithm breaks down the query sequence into smaller subsegments called seeds. The default length of a seed is usually 3 or 5 residues long, but it can also be adjusted. These seeds are then compared to the sequences within the database. As a database matches the seed, the alignment gets extended in both directions. This produces a local alignment that continues until a maximum scoring extension is achieved. The use of seeds for initial matching significantly increases search speed, and the resulting alignments only miss a small number of significant matches. After the algorithm has determined local alignments from all possible arrangements of sequences, it evaluates whether the degree of match is actually justified by biological significance or whether it is due to chance. The BLAST algorithm therefore determines the expectation value, or E-value for short. This determines a number of matches we would expect by chance. The lower the E-value, the better is the alignment between the query sequence and the found sequence, also our target sequence. For an example, a value of 1 would mean that a random match of similar length and percentile identity would be expected. A meaningful cutoff value should always be determined based on your research objective. For DNA sequences, it is recommended not to go higher than e to the power of -6, and for protein sequences, you should not go higher than e to the power of -3. If you want to learn more about the BLAST algorithm, I would recommend reading the publication, or as shown in our first lecture, taking advantage of the training opportunities represented at NCBI or EMBL could also help. Before I finish this lecture, there is one more type of algorithm that I have not covered. These are machine learning algorithms. Machine learning algorithms are used in many modern alignment tools, such as tCoffee, Clustral Omega, or Hammer. They often use a statistical method to predict the probability of future events called hidden Markov models, or HMMs for short. HMMs provide the basis for a number of complex models that can be used for multiple sequence alignments, profile searching, or sequence detection. I will not cover the complexity of these methods in this lecture for good reason. The reason is that this is an introductory course in computational biology and not an advanced course in algorithmics. However, I think it is important that you have at least heard about this topic. If you want to know more about it, I recommend the shown tutorial by Rob Finn, where he explains the EMBL EBI Hammer algorithm and its applications in detail. That's it for today's lecture. If you need more information on the topics discussed, or if you want to study them more in depth, check out the supplement to this lecture. I've included recommendations for books and other resources. In our next lesson, we will apply the knowledge of this lecture to work with various web and software tools. So I hope to see you again in the next lecture.